All right, we're going to continue on with our review for cells and cell processes. This is part two. We were in the middle of question number 15 on your study guide. So we were talking about the movement of CO2. CO2 is a little molecule. It can pass across that semi-permeable membrane, doesn't require ATP, so that would be like this example over here. Movement from high to low concentration. Also, high concentration on the left, low concentration on the right. That's an example of diffusion. It's passive transport. However, movement opposite to diffusion, from low concentration on the left to high concentration on the right, in order to go the wrong direction, the cell has to use its energy. It has to push those molecules to the other side. We call that active transport. Anytime the cell is using energy, it's active. Facilitated diffusion is a super hard word. Facilitated means helped, and diffusion means it's passive. It means it's not using any energy. So this on the diagram is an example of facilitated diffusion. It's where a protein is being used like a hallway. The molecules are traveling through, but they're not being pushed through. And so no ATP is being used. And so that's an example of passive transport. In the case of large molecules, we're on letter G right now. Large molecules also have to be pushed across using some energy, and that's active transport. And then the sodium potassium pump, we saw a video about that, and let's see if I can remind you this. Give it just a second to click. There we go. This is the sodium potassium pump. We saw a video about how the pump opens and closes like this, and molecules are allowed in and out or across the cell membrane, but ATP has to be used, and you can see that in the diagram down here. So that's an example of active transport with the sodium potassium pump. All right, now the question is, what's the difference between exo and endocytosis? So here's the slide that's about it. Exo means it's exiting out of the cell. Endo means it's coming into the cell. So exo and endocytosis are both forms of active transport. So it uses ATP in both circumstances. Okay, then it asks about what happens if an animal cell is placed into a hypo or hypertonic solution. That's from this previous set of notes, and I just want to remind you where we found them right there. Isotonic, hypotonic, hypertonic. With hypotonic, you're going to remember hypo-hippo. So if the cell starts to swell or the pressure inside of it increases, that's a hypotonic solution. If the cell shrinks, like what would happen if you were drinking salt water, then that's a hypertonic solution. So let's see what the question said. What will happen to an animal cell placed in a hypotonic solution? And what happens if you place a plant cell in the same solution? Well, water is going to rush in, and if it's an animal cell, that animal cell is going to swell, and eventually it's going to pop. A plant cell has the advantage of having a cell wall around it, and so the pressure is going to build up. It's bad for an animal cell. Animal cells don't want high pressure, but plant cells like that high pressure because it allows them to stand upright so they can photosynthesize. That's called Turgor pressure, and it's good for the plant. All right, number 18, what's an example of a hypotonic solution? Distilled water. What's an example of a hypertonic solution? Ocean water. If you drink the ocean water, it pulls the water out of your cells and it causes you to be dehydrated. Okay, next we talked about the eggs and what happened in that egg video.
What happens to a cell when it's placed in a hypotonic?
I had this all keyed up, or all, what's the word? Fast forwarded for you, whatever. I had it on just the right place for you. There we go. So remember that when the woman placed the egg into a hypotonic solution, which was just distilled water, the egg got really, really big, hypo-hippo. And when she placed it into corn syrup, which was very thick and had very little water in it, that's a hypertonic solution. The egg shriveled up and it got a lot smaller. I think that's about it. Let's move on to the Osmosis Demo Lab. So we, in class, we looked at potatoes that were soaking in salt water and in distilled water. The potato that was in distilled water was super rigid. It was really hard for you to bend, and that's because there was so much Turgor pressure. The water was inside those cells, and they were rigid with the cell walls because potato is a plant. So they felt really firm. Whereas the onion cells, this is a good photo of the onion cells. Normally the onion cells in distilled water or regular water, notice that the the cell is completely full of the pink dye in this circumstance. But then when we put it in salt water, the water gets sucked out of the cells and they all look all shriveled up. There's still a cell wall there. It's hard to see, but the inside of the cell shriveled up, and that lowered the Turgor pressure. A plant that was trying to grow in that circumstance would be all wilted. It would be all tipped over because it has that low Turgor pressure. All right, I'm going to move my picture here out of the way. Did the onion cells in distilled water have high or low Turgor pressure? I'm in question 23, and the answer is high. It's good for plants, bad for animal cells. And then finally, the semi-permeable membrane lab. Let's see if I have the picture over here. There we go. You remember that's the one we referred to as the Jolly Rancher. We put sugar and starch solution. So the sugar would be represented by these little red dots, and the starch would be represented by the big blue dots. The semi-permeable membrane, it meant small things can pass through. So the sugar molecules were able to pass out, and the starch molecules were trapped on the inside. We tested the water on the outside to see what had escaped. Testing the water on the inside doesn't help because they're both in there. We needed to see what got out to figure out which one was smaller. And then the interesting thing that happened in our lab is that when we added iodine to the outside, the iodine actually was small enough to get inside the Jolly Rancher, and it touched those starch molecules and it turned blue. And that's why the inside of your baggies turned sort of a blue color. All right, ladies and gentlemen, that is it. That doesn't mean that you're all ready for the test, but that's a quick little 20-minute review. And I really suggest that you take a look at the study guide, make sure you know it inside and out, and that you take a look at the Quizlet and practice those flashcards. The quiz on Quizlet, not hard enough. That's too easy. But the flashcards on Quizlet are really good. All right, have a good night and good luck on your test.